welcome to Virtual Seminars Precambrian Geology. We're excited today to have Dr. Heda Agic presenting for us on insights into habitats of Neoproterozoic eukaryotes from single fossil carbon isotope measurements. And next week we'll have Dr. Robert Stern to give a talk that follows Dr. Condi's talk from last week quite well. It's titled Disruption of a Mesoproterozoic Stable Lid Tectonic Regime and Establishment of a Global Plate Tectonic Mosaic Throughout the Neoproterozoic Time. So looking forward to that. At the end of today's talk, just like as with any week, we will have a question session. And during the question session, I ask that you either raise your hand by typing an asterisk in the chat, or you can, if you do not want, and if you type an asterisk, I will direct you to unmute yourself and ask your question. If for some reason your microphone is not working or you would rather not ask your question personally, then you can type it into the chat and I will ask it for you. We'll address these questions in the order they're received, and I will, unless I direct you to unmute yourself, please do stay muted throughout the entirety of the talk. So with that, I'll go ahead and introduce our speaker for today. So Dr. Agic is a paleontologist mainly focusing on the evolution of early eukaryotes from the origin of a complex cell through the origin of multicellularity. She receives her undergraduate and master's at University College London in the UK, for which she studied Neoproterozoic Cambrian phosphatic microfossils from Mongolia and China. She then studied at Uppsala University in Sweden for a PhD on palaeobiology and diversification of Proterozoic Cambrian photosynthetic eukaryotes. From 2014 to 2019, she was also a project scientist with the Tromsø University Museum working on the Ediacaran Cambrian transition in Norway. The work she's going to discuss today is part of her current postdoctoral fellowship with Dr. Susanna Porter at University of California, Santa Barbara. This project on organic walled microfossils from the Neoproterozoic Chuar Group involves both paleontological and carbon isotope data on individual microfossils and seeks to help reconstruct early eukaryotic ecosystems. So with that, I'm going to leave Dr. Agic to tell us all about it.
All right, well, thank you for this invitation, and also thanks to Alex and Andre for organizing this seminar series. I really enjoyed it so far. So today I'll talk about Proterozoic organic walled microfossils, which are our prime archive to investigate early evolution of eukaryotes. And there are a lot of unknowns about this group of Precambrian fossils, but here I'll show a novel approach, which we can kind of squeeze out paleoecological information. And this is work I've been doing in this postdoc with Susanna Porter, Phoebe Cohen, and Chris Junium with NSF funding. All right, to put things into a temporal context and to show you the fossils that I'm excited about and the time I'm excited about, let's briefly go over the timeline of the Precambrian history of life, time on the x-axis. From the oldest fossils in the Archean to the start of the Phanerozoic, which is mainstream and so not included here. And Precambrian represents 88% of Earth's history. And during most of this time, in terms of the fossil record, we're dealing with soft-bodied forms. And the oldest known body fossils appear around 3.4 billion years ago. And then eukaryotes, which are organisms with a complex cell, evolved following the Great Oxidation Event. And the oldest eukaryotes are preserved as organic walled microfossils from China. That's around 1.65 billion years ago. And they remain mostly single-celled until 1 billion years ago, when we see multicellularity in a few clades. Oh, and a cool paper from Maloney et al. Just came out in geology as well. So we have more and more evidence of this onset of multicellularity around this time. And then the first visible life, Precambrian's charismatic macrofauna, the Ediacarobiota, as well as quite young, evolving at the Ediacara and Cambrian transition around 500 million years ago. So Proterozoic is a time of microscopic life, and on this micro scale, we see one of the most important evolutionary events for us, which is the origin of a eukaryotic cell. Eukaryotes are traditionally one of the 3 domains of life, and they differ from their bacterial relatives, bacterial and archaea relatives, in having kind of a larger and a complex cell with a nucleus and a suite of organelles that perform specific functions, for example, mitochondrion or plastids that have pigments for photosynthesis. This group includes macroscopic life, like this whale here, and animals, plants, fungi, algae, but also a huge diversity of single-celled forms, collectively called the protists. And this is a very busy phylogenetic tree of eukaryotic groups, but it is useful to illustrate the diversity, especially of microscopic forms. But eukaryotic relationships are actively researched, and their fossils are an enigmatic group, and there is much we don't understand about deep time eukaryotic paleobiology and palaeoecology. I'd like to spend just a few slides here to introduce the group of fossils that are kind of my primary research subject. So they have a lot of names, but the most useful descriptor for these is organic walled microfossils because they're small and soft-bodied and preserved as organic matter, mostly within fine-grained siliciclastics. It means they do not have skeletal elements. That is kind of a later innovation. And some are instead composed of aliphatic biopolymers, some similar to sporopollenin in modern plant spores. Others are not as sturdy, but can still be preserved organically if deposited under favorable conditions. This is a functional group for most part. Their phylogeny is not resolved. They likely represent a variety of persistent organisms, meaning that they're polyphyletic. Narrowing down their affinity is hard, and we'll come back to that in a second. And importantly, we use a few criteria to tell apart eukaryotic and simpler bacterial cells. So eukaryotes are larger, usually. They have a complex vesicle morphology with these sturdy spines. And bacteria are not able to produce so because a cytoskeleton is needed in order to form a complex cell morphology. And then also some cells have reproductive structures called the openings or the excisement, and they generally have these sturdy walls. They're commonly found in fine-grained siliciclastics, mostly mud rocks. We extract them from the rock matrix by macerating them in hydrofluoric acid, which dissolves the silicates, then boiling them in hydrochloric acid to remove carbonates and fluorides, and then filtering them through usually 10 to 25 micron meshes. Filtered residue can then be mounted onto glass slides for microscopy. Light microscopy gives us an overview of who's there, and it's useful for quantitative studies in ecology. And scanning electron microscopy is useful for detailed observations of anatomy and kind of gives us the high-res structures. And we care about this group because organic walled microfossils represent the first complex life until the onset of the first visible life, like the beautiful Ediacarans later on. But we know very little about them, like, when did crown group eukaryotes appear, how did they live, and so on. Morphology only gets us so far. So for instance, these spines on this form here. So spines or processes occur in different persistent clades and have different function. And so you can find literature describing how they help phytoplankton stay in the upper column by preventing sinking, but other groups, they're also used by benthic organisms to prevent from burial and sediment damage. And so we still know very little about their paleoecology, like habitats. Morphology only gets us so far, which then makes it difficult to directly test hypotheses about the role that environmental change may have played in eukaryotic evolution and diversification. A big question in eukaryotic evolution is the effect of changing ocean oxygenation in the Proterozoic. Midway through Earth's history, the planet was quite different, and here we have oxygen levels from a compilation of proxies in percent of present day levels on y-axis through time on x-axis. And oxygen remained low for about 1 billion years from the Great Oxidation Event until later, until the Neoproterozoic, and even, as you've seen recently, through early Phanerozoic. Yet, tiny eukaryotes show up around this interval of time before the second rise in oxygen. But commonly, the low oxygen levels were invoked as a brake on the evolution of complex life. And while that is mostly true for macroscopic and mobile life, the fossil record through this time is quite rich in these diverse groups of microscopic eukaryotes, even multicellular and biomineralizing forms. And a lot of this diversity was discovered in recent years. This is just a flavor. And there's accumulating evidence that by 1 billion years, we have crown groups. It's assumed that early eukaryotes also needed oxygen because they possess mitochondria for aerobic respiration, and they do have higher energy requirements than most bacteria. But really not knowing exactly, what environments they inhabited or where in the water column makes it challenging to test any hypotheses about early eukaryotes. As I said, so all eukaryotes today, apart from oddballs like weird derived exceptions, have organelles like mitochondria that are used in respiration and would require oxygen to some extent. But it's still pretty unclear when that acquisition took place. We can assume that our microfossils had them? So the last eukaryotic common ancestor, or LECA, shown here in blue, probably possessed mitochondria, as we know from molecular biology. But there would have been lineages of early eukaryotes, members of their stem groups, that were more adapted to the prevalent anoxic environments at the time, and those would also have entered the fossil record. So in part, to test where and how early eukaryotes lived can be done with the fossil record by analyzing conditions in which the microfossils would have lived. So we can look under what conditions our fossil death assemblage was deposited in, and so we can do so by using proxies like iron speciation, which tell us about local redox conditions of the bottom water in which fossiliferous shales were deposited. But there are a few scenarios to consider here. So early eukaryotes could have inhabited water column with oxic or ferruginous bottom water. They could have inhabited like a fully oxygenated water column, but also we see microfossils occur in sediments were deposited in anoxic shales. And so another possibility is that they could have maybe lived in such a condition. And so in the second scenario, there are a few things to consider. So fossils that get deposited in our death assemblage may represent several things, not necessarily just the bottom water. And so they could have lived, they could have been maybe restricted to this surface that was presumably oxygenated, or they could have lived, just at depth, which was often ferruginous, or they could have lived kind of everywhere. And so perhaps some organisms were following a chemocline, like ciliates or deep green algae in modern fjords. And so to get more precise information from our anoxic samples to resolve between those cases, we must somehow distinguish between them. One approach that can act as bathymetry and tell us more about this is to explore what carbon pools influence our fossils. So, for example, we may see environments with a carbon isotope gradient, and those would be cases of an active biologic pump with phytoplankton producing at the surface, and or a case with anoxia at depth and thus less mixing. This will lead to different isotopic signals in different parts of the water column, and we would expect heavier carbon isotope values at the surface, due to export of light carbon from surface to depth via production, photosynthesis. And if there's a lack of circulation or a lot of production, then the heavy carbon would be accumulated at the surface. In contrast, in a well-circulated ocean, there would be a smaller gradient. But we would still expect, the organic matter at depth to be isotopically lighter because it derives from DIC of respired carbon. And for example, this was shown for organic matter from cryogeny and ediacaran from Australia, where kerogen and biomarkers derived from microbial mats here were found to be depleted in δ13C relative to planktonic material. So with this in mind, we can try and assess this for Precambrian ecosystems by measuring carbon isotopes of single microfossils, just to see if we see an isotopic gradient, to see if our fossils have isotopic composition that would reflect a particular part of the water column maybe, or if their values are simply a mix, simply reflecting the bulk organic carbon. And Phoebe Cohen and Chris Junium are applying this approach to Devonian microfossils to study carbon cycle dynamics through the late Devonian biotic crisis. So keep your eyes open for that. So to gain this info about palaeoecology of Neoproterozoic eukaryotes, material was collected from the late Tonian Chuar Group, which is exposed in the Grand Canyon in Arizona. Chuar spans about 1,600 meters of mostly mud rocks, with some sandstones and some dolomites. Its age has been constrained to about 770 to 729 million years, so this is a quite late Tonian period, before the snowblower glaciations. This locality has yielded a lot of paleontological information through decades since the discovery of carbonaceous compressions Chuaria by Walcott in the late 19th century. And diverse and beautifully preserved organic wall microfossils occur throughout the Chuar. And these include quite a few eukaryotic taxa that are distinguished by processes or envelopes or surface patterns of some kind. And then another microfossil group appears at the top, and these are vast-shaped microfossils and likely remains of testate amoebae. Out of the Chuar assemblage, prokaryotes are quite common. There are several filamentous and cell aggregate taxa, like Rugosopsis and Polytrichoides. And these are generally interpreted as components of a benthic microbial mat. This comes in super handy here. So I know where these guys lived, and so they can provide me with some orientation of what is going on at depth. And they tend to be isotopically light, which is consistent with what you would expect in a system with isotopic depletion and depth, and I'll come back to that in a second. Right, so sediments and microfossils were deposited at various depth regimes, seen in several regression intervals here. And under different redox conditions of the bottom water, oxic or ferruginous, and then some euxinia appears at the top here. So we can look at what's happening at different depths and in different redox scenarios. Redox and depth don't appear to affect microfossil preservation all that much, or even their abundance. And so recent work from Tina Woltz in our lab show that they are marginally better preserved in oxic samples when it comes to recognizable surface pattern. And there isn't a lot of difference with preservation and depth. And this is out in geology, and I encourage you to check it out. Okay, and this is a carbonate carbon and organic carbon isotope profile through the Chuar Group from Carol Daler and colleagues. And this is mostly δ13C org because it's mostly shales. And this record varies a bit through the Chuar and then undergoes this positive carbon isotope excursion in the Awadabi member of the Kwagund Formation. And I've also selected samples from here to see if our microfossils would show that excursion. Okay, so microfossils were extracted from shales throughout the Chuar succession. This was done with a modified palynological acid maceration approach to minimize contamination from external carbon sources, and we can discuss all details about that later. Here in the middle is acid-insoluble residue that is left after maceration. A mix of organic walled microfossils, bits of microbial mat, and clumps of fluffy kerogen. And then out of all of that, individual microfossils are hand-picked in a laminar flow hood and placed into these silver cups for analysis. Shy of 200 microfossils were analyzed with a nano-elemental analyzer, IRMS at Syracuse in Chris Junium's lab. And this follows a procedure for analysis of very small quantities of carbon, down to 20 nanomole or so, which amazingly allows us to combust and analyze individual microfossils, some of which represent single eukaryotic cells. And microfossils come from a variety of settings, as I mentioned, that are present in the Chuar, and samples representing deep and shallow environments. And as well as oxic and ferruginous bottom water. And only low TOC shales were analyzed, as those were found to yield best microfossil preservation, which was shown by Tina Boltz. And I should note that only thin layers of shale were processed whenever possible, so that time averaging bias is minimized. And importantly, the microfossils here record the primary organic carbon. So another goal we have with these analyses on Precambrian organisms is to unlock environmental information hidden in them. And so before, it wasn't really possible to utilize them, to the same extent to which Phanerozoic microfossils are used, like forams or dinoflagellates. And this is why this approach can be so cool. And here are carbon isotopic values of different organic carbon components measured in the Chuar group. So height is here on the x-axis, and δ13C value of microfossils on the x-axis. Sorry, height is on the y-axis. And they can be remarkably, so the fossils can be remarkably offset from the isotopic value of the bulk acid insoluble mass rate in these red squares, and the average microfossil value, which is shown in these crosses here, is also usually offset from the bulk mass rate. And then the bulk mass rate itself in red is offset from the bulk rock δ13C org shown in blue triangles, and the individual microfossils also aren't reflecting the bulk δ13C org value. Okay, remember that positive excursion in the Awadabi member from a few slides ago? The bulk rock data in triangles measured by Daler et al., and they shift towards more positive values at the top of Chuar, but the microfossils are unfazed. They have this huge spread of values. Now, this shows us that the microfossils do not follow a secular trend of bulk carbon isotopes. Instead, I would argue that this range suggests that they probably incorporated carbon from various pools. Here we plotted these components in a box and whiskers plot. If we compare isotopic values of bulk shale, bulk macerate, and individual microfossils, they're not that different, but fossils have a very broad range, both heavier and lighter than the bulk. It could be that some fraction is perhaps being lost in maceration, like bitumen, but this range could also be due to microfossils having heterogeneous sources of carbon or doing very different things metabolically that are then affecting their isotopic composition. So why are they so different? Are there taxonomic differences? Again, stratigraphy is on y-axis and δ13C on x-axis. Each color here represents a different genus of microfossils, including our mat builders in green. And this is a bit chaotic to read, but that's kind of the point. There is a lot of variation among taxa, even within taxon, and each of these lines is a horizon, so a macerated shale sample, and contains a bunch of different taxa, and they're showing pretty different isotopic values. That's up to 22 per mil per sample, on average 12 per mil. And taxa also deviate from bulk values of the mass rate in black squares by up to 15 per mil. It can be both enriched and depleted relative to the bulk. So this variation again suggests that individual microfossils could have taken up carbon of different values. Okay, now out of these individual microfossils, let's single out some. Here are values of mat pieces and mat builder taxa in green, and they're mostly hanging out here on the left-hand side among these sort of depleted values, and they're generally lighter than the bulk, and they can be lighter up to 13 per mil than the bulk, and on average they're lighter by 4 per mil. Then among enriched microfossils, there are envelope-bearing Simia and the Secanthamorph Germanosphaera. Simia is in purple and Germanosphaera in pink. And okay, so Simia does have a few outliers, but generally both of these taxa tend to be enriched. And Simia is interestingly enriched relative to the bulk in 11 out of 13 samples that it occurs in. And it's usually heavier than the mats, quite heavier than the mats, on average by 7 per mil, and this can be up to 13 per mil. Okay, so for a lot of taxa, even within taxon, there is quite a bit of variation. So the broadest range of isotopic values is seen in Leosphaeridia. And this is exactly what we would expect. So Leosphaeridia are smooth spheres present throughout Proterozoic, and they're likely polyphyletic, meaning that the simple form actually represents a variety of biological species, and this shows us that's true. Different biological species that dwell in different parts of the water column would have incorporated carbon of different range of values. So if our entire assemblage, if our entire death assemblage is recording some short-term environmental change or temporal change, then we would expect huge variation in all taxa, regardless of affinity. But some distinct Tonian eukaryotes, like brainy Cerebrosphaera or Squamosphera, actually have a narrower range of value within taxon. I also compared depth and redox of the analyzed samples. Symbols here represent different depth and oxic, and then here we have in these horizons, we have oxic setting in blue and anoxic in orange. And there isn't a lot of difference in the spread of values between oxic and anoxic samples, but shallow samples, particularly shallow oxic samples, or peritidal samples in circles, have a narrow range of fossil carbon isotope value. Now, perhaps this suggests that the water column in such settings wasn't really stratified, and so we wouldn't see an isotopic gradient. And then the broadest range of values is seen in deepest anoxic samples. These are distal subtidal settings. And this could reflect a stratified water column, which then also gives us a range of water column habitats that could have been inhabited by the Chuar microfossils. Okay, here we're only looking at Leospherids, and I know that they're the most boring microfossil, but this is, I guess, their chance to shine because they're abundant and, remember, polyphyletic. And so let's have them here as to represent a subset of the Chuar assemblage. So x-axis is offset of the carbon isotopic value of our microfossil from that of the mat in the same sample. And it's coded by depth. So in the deepest, distal subtidal parts of the basin in purple, the Lewis ferrites can be quite offset from the math value, and they show a lot of they show quite a bit of a range here. But then in shallow environments, in shallow in these kind of peritidal environments in green, their values are actually pretty tight, which again means that the water column in these shallow oxic samples probably wasn't stratified enough to get some kind of a gradient. Now, let's zoom in on a few samples to see what is going on in individual horizons. So, within one single macerated sample. In these deep water samples, we have anoxic on left, oxic on right. There is some variation within taxon, especially among these polyphyletic leospheroids, as I've said. But these mat builders in green are lighter than the bulk macerate d13C. Chuaria is also quite light, lighter than the macerate. But some taxa, like Simia, are enriched, and Simia is the cell with an equatorial envelope. And then some well-defined eukaryotic species show similar values within sample, like Cerebrosphaera, or this common bubbly taxon in the Tonian Squamosphera, or process-bearing Linolithosphera. In shallow water samples, again, we have anoxic on left, oxic on right. There's a bit less diversity and also less spread. And most taxa are heavier than the bulk. And here, Simia isn't just enriched, but it also shows a spread of values. And it's not super offset from the mat builders anymore, especially in this shallow oxic setting. Now, going back to the question about early eukaryotic ecology, where do they live? What do they do? I'll simplify this with a water column cartoon. So, maybe what it would have looked like in the Chuar basin. So we know that the mat builders, the squiggles, live at the bottom. And so in this example, they're lighter than the bulk organic carbon. On average, they're around -28 per mil, and they tend to be lighter than the bulk. And then Simia can be pretty enriched, around -15, -14. On average, through the Chuar, its value is -19 per mil. And through the Chuar, Simia is heavier than the mat builder values on average by 7 per mil. So presumably, it incorporated its carbon in enriched and likely oxygenated surface waters. In any case, perhaps away from the mats. Interestingly, in oxic samples, Simia has a broader range of isotopic values, as I've shown, not necessarily reflecting just the enriched carbon pool. So maybe it can hang around a bit deeper. So this is one taxon that we mainly see in the presence of oxygenated water. On the other hand, things like Chuaria and especially Squamosphera are consistently light, quite close to the mat value. And this is supporting, some previous information back from 20 years ago or so of Chuaria as a benthic taxon. So either as a colony, as Martin Steiner proposed, or a resting cyst from the Kumar paper. And the ornamentous Squamosphera, this shows us was probably also benthic because it's close to that mat value. Some other common eukaryotes like Linolithosphera are more depleted than the bulk and also offset from the mats. And so it is less likely that those lived at the surface. Now, these are still preliminary results, and I'm picking more fossils for better statistical confidence, but there is a pattern here. And this indicates that single-celled eukaryotes could have inhabited perhaps different parts of the water column. I mean, not all species that we see here are enriched, and so we cannot assume that they were all restricted to oxygenated surface waters. And instead, this is a bit more indicative of habitat heterogeneity. And this is similar to some modern protists that can thrive in deep anoxic environments or fjords. And our enriched simia could have been, near the surface or preferring oxic environment. Other explanation for its heavy values could be that maybe it used some kind of different metabolism, like bicarbonate pumping. But I'd like to say that for a long time, simia was suggested to be related to presinified green algae due to its similarity with their cysts or phycomas. And I think that perhaps its little equatorial envelope here helped reduce its sinking rate and maybe stay in the water column to happily photosynthesize. And so considering all of that, this planktonic mode of life in the upper water column is not surprising for simia. So this approach may offer clues about its affinity and generally about affinities of the other organic wall microfossils, and we can learn more about these, about this enigmatic group. All right, so some of the main points from these initial results are that the microfossils that we have picked so far show a broad range of isotopic values that are usually quite offset from the bulk acid insoluble organic carbon value. And individual species are showing different value ranges. And this kind of confirms some of the previous concerns that perhaps we shouldn't trust the bulk carbon acid value that much, which is showing us just kind of an average of a given water column. And this has been suspected for some time. Classical literature has discussed how isotopic records of bulk organic matter can be quite difficult to interpret because they would be coming, they would originate from contributions from sort of various sources of organic carbon within sediments, which can have heterogeneous isotopic composition. And the microfossil record is supporting this to this point. As we saw, they didn't follow that positive carbon isotope excursion in the Awatabi. They're quite heterogeneous, and which in large part that probably reflects their ecology. And I'll finish on a positive note sort of by saying that this is pretty exciting because there is some emergent pattern. We're able to get some information like the striking distinction between these depleted mat builders at depth and enriched microfossils that were long assumed planktonic. And hopefully with more such studies, the microfossils may come to play an important role in helping us understand not only eukaryote palaeoecology in the Precambrian, but also environmental variation and Precambrian carbon cycle dynamics. And lastly, thanks to my collaborators and thanks to NSF for funding, and fossils were collected from traditional Kaibab and Havasupai lands, and we're thankful for permissions to collect. And lastly, thank you all for listening, and I'm looking forward to any thoughts or questions that you may have.
Hey, thank you very much, Heda. That was awesome. I think I really enjoyed seeing this data with the reference being the map builders. I thought that was really interesting. You have a couple of questions already in the chat. The first one from Nicholas Christie Blick saying, fascinating. Given the huge variation in values, what is the significance of these data for chemostratigraphic curves?
Well, that's the question, right? If we can't, so for chemostratigraphy, we've been using a lot of carbon and organic carbon to a lesser extent. But that's the bulk value of what we see in a given sample. And usually, we go out in the field and we take these samples, and they do represent, as I said, a degree of time averaging. Because our sediments and our microfossils are going to be accumulated over a period of time. And so the microfossils themselves are giving us kind of a narrower window into what's happening, because the microfossils, we still don't really understand what's their lifespan, and we can assume that it's pretty short if we look at modern protists. And so they would have actively taken up carbon over much shorter time than your average sample, and they're probably kind of a better reflection of what's going on at a time than just an average bulk value. And so in this case, in the water bee, there could be another component that is influencing that positive carbon isotope excursion. But the microfossils are not reflecting that.
All right, our next question is from Adriana Rizzo. Adriana, if you'd like to unmute yourself, go right ahead.
Hi, Heda. Thanks so much. It was a great talk. It's interesting to see how this is progressing. I had a question about the taphonomy of organic walled microfossils. To what extent is it, you say that they represent primary organic carbon. To what extent is it known, and is there sort of about them being, are they primarily sourced from sort of the preservation of existing recalcitrant organic structures, or could there be a role for templating of structures that sort of are degraded and replaced with kerogen? And is there sort of any, you know, actualistic studies that have looked at this?
In terms of actualistic studies, not really. In terms of kerogenization, to some extent, most of these are kerogenized, or at least that's assumed, that are mostly kerogenized, but not to a great extent. So sometime, and for that we usually use the thermal alteration index following Heda et al. 86. By sort of looking at the color of the microfossils to infer their thermal alteration. And most of these have these sort of light values, and they haven't undergone high thermal alteration. And most of the highest temperatures, paleogeologic temperatures that have occurred in the Chuar are at the very top of the formation, above the Awadabi, but those samples, and they're pretty euxinic as well and very rich in TOC, but those haven't yielded microfossils. And so this material is pretty uncooked. And, but yeah, we're not sure about their individual composition. This is something that we're looking into right now. A colleague of mine sort of zapped Chuaria with XRF to see what's going on and how that is preserved. But so far, most of the studies have been done with FTIR characterization of what's happening, and most of the Precambrian microfossils are composed of these sort of aliphatic biopolymers. But honestly, I don't know what's original and what is diagenetic in this case.
Okay, thanks. One way of getting that is you might want to look at a similar sample size for the bulk and microfossils to see if they have the similar spread, because it looked like there was more spread in the bulk, but there was also a lot fewer measurements for the bulk compared to the microfossils. So.
Yeah, that's a good point. That's something. Yeah, thanks.
Great, thank you.
Right, and it looks like in the chat, Phoebe Cohen had something to tack on, and she said, Adriana, we also have lots of detailed SEM, TEM, etc. analyses that show the primary nature of the wall structure itself. And then our next question is from Gregory Vitalik saying, lovely data, Heda. 22 per mil is a lot, most comparable with fungi rather than primary producers. Fungi, of course, feed on a variety of sources, including sulfur bacteria and methanogens. So it looks like that was a comment rather than a question, but.
Right, so the carbon values that we get in these things, like obviously, okay, to some extent, you are what you eat plus minus a couple per mil. And so what you eat is going to be a part of your carbon isotope composition. But this is not the only thing that's also affecting the carbon isotope composition. And the value, the bulk value of the sample of the water column that you would have been in, and how you're offset from that value is perhaps more diagnostic as to what the organism is doing rather than just raw isotopic value, because you're taking up your carbon in a system. But also, you know, minus 22 per mil, that's, if you look at the Shidlovsky diagram for metabolisms of different microbial groups, not just eukaryotes, but also algae, methanogens, and so on, there is a lot of overlap between a lot of these groups. And photosynthesis is quite, you know, has quite a chunky range of different values that it yields. And these very, very light values from methanogens and so on are also pretty broad. And there's a lot of overlap there. So that's not very helpful for understanding what those microfossils are doing metabolically.
And so Christopher Junium does add, saying, our 13C analyses of individual kerogen grains don't show anywhere near the range of variability that we see in the fossils. So I think that is connected to what Adriana was mentioning. Does anybody else?
Yeah, the kerogens are quite, sorry, the kerogens are quite close to the bulk value.
Okay, do we have any more questions for Dr. Agic?
Yeah, maybe I can ask Heda. So just thinking, so it seems like you have two possibilities that you discussed. One is different metabolism, and another one is depth gradient. So thinking about your data around minus 15, minus 10, it seems like it would be hard to explain it just by metabolism, because it would be a very small fractionation. And sorry, not by metabolism, by depth gradient, because it would be very small fractionation. So it could be different metabolism, but when in the same fossil you have such a big range, so I guess maybe it's possible to switch from one metabolism to another in the same organism. I don't know. So my two questions, like when you prepare, when you macerate, do you use any organic solvents? So is it possible that you introduce any contamination with carbon when you do it, or do you analyze the standards that you analyze with the same size as your samples? And the second question come in, like in Russia, when they did microfossil biostratigraphy, they common saw intervals that seems like recycled microfossils. So it would be microfossils that were eroded from pre-existing rocks and introduced, and which should be quite possible if you have low atmospheric oxygen. You could potentially transport organic carbon and introduce it into new sediments. So could it play a role in what you observe?
Right. So reworked microfossils are quite a problem for this, but we I don't pick those, so they're actually not that difficult to single out in your given sample. Because because usually they're going to have a little bit of a different coloration and preservation than majority of the microfossils within that sample. And obviously, like that, the color and preservation does change, you know, in a single macerated sample. But usually reworked microfossils will stand out, and they will be generally more poorly preserved. And if something is really poorly preserved, I won't pick that. And I worked a lot on in sort of these early Cambrian units where reworking is quite a bit of a problem, and the reworked microfossils are quite easy to recognize, and so they can be avoided. It's not something that I have observed in the Chuar so far, so I don't think that there would have been migration of older microfossils. And then your first question, I think, was about processing. So for this, we have modified the typically used acid maceration method. And that's done with hydrofluoric acid and then hydrochloric acid. And usually these residues in palaeoecology are stored in ethanol and acetone. But those have been found to introduce quite a bit of bias, and they give a bit of an offset in carbon isotopic composition. And so for this, we've stored them in methanol, which kind of has the least effect. Methanol and dichloromethane have the least effect on that carbon isotopic value. I guess they don't get absorbed. And sorry, what was your first question? I think it was you had three questions.
It just was a comment that if it's metabolism or if it's the gradient, and it seems like values don't exactly fit to either of them.
Right, and so okay, yeah, there could be different explanations for this isotopic heterogeneity that we're seeing. And maybe this is going to be a bit of a circular argument, but like if we're looking, for example, if we assume from previous paleontological studies and from morphology that some of these are photosynthetic, then we can assume that our mats are going to be, you know, they've been interpreted for decades now as cyanobacteria. And so if we assume that they're photosynthetic and perhaps, you know, these, if we assume, again, based on morphology, that maybe our simia is photosynthetic and they do have this like huge offset, then perhaps if they're doing the same metabolism, maybe this is telling us they're living in a different place. But yeah, this is something to tackle in the future.
So I guess it's possible you went more in depth than this in your talk than I'm remembering, but I thought I was just sort of having, these death assemblages and shales are just kind of where you have things preserved, I mean, obviously where you have things preserved rather than where they were living. And you're getting these from shales, but you also have these mat builders and you have other things occurring in the same shales. So they're clearly just being sourced from all sorts of places. Did you have primary mats, and did you get fossils from primary mats?
There are laminated shales in the Chuar. So yeah, I would assume that they're primary.
They're primary mats, and then these other fossils are preserved with those mats, the non-mat builders.
And so with mat builders, by mat builders, I mean different taxa that are either filaments or cell aggregate taxa that are sort of, whose taxonomy is well known, that were interpreted as mat building cyanobacteria. And by mat pieces, those would be these kind of fluffy clumps of organic matter that have filaments kind of sticking out of them. They're quite easy to pick because they're you know, a bit big. And so these are pieces of the mat. But yeah, also laminated. You can see that lamination in shales as well.
Okay, so, and I guess I don't have much of a question or even a comment. I was just wanting to think whether or not these mat builders were definitely from in place or from somewhere else, and the other fossils could, I mean, I guess they could possibly be from environments that do have a significantly different carbon isotope composition of the environment too, right?
Right.
That isn't necessarily, I mean, I know we're talking about gradients and stuff, but I mean, is it possible, do we have environments that do have significantly different carbon isotope composition that isn't related necessarily to their metabolism that they could be transported from?
So the beautiful thing about the Precambrian, which is why I love it, is that we don't have to worry about the pesky terrestrial environment and the input from the terrestrial environment that much. I mean, at this point, there could have been definitely, and there is evidence for life on land, probably in terms of like cyanobacterial pools and stuff like that. But yeah, I'm not sure how much of that input would have. That's another possibility, that the influx of that input would give us that isotopic heterogeneity that we see in the Chuar. But what's important here is that maybe we would expect more heterogeneity than in shallow proximal environments that are closer to the terrestrial realm, but actually in those samples, we're seeing less heterogeneity. Most of our heterogeneity is in deep water, which is proximal subtidal. And so in that case, I don't think that there would be a lot of input from terrestrial organic matter.
Okay, that makes sense. Interestingly, in a few weeks, we'll have Chuhai Zhao give a talk on terrestrial life in the aftermath of a snowball Earth. So it will be interesting. We have a next question from Sarah Slotsnik. Can you speculate more on the difference between bulk shale and bulk macerate?
Okay, so bulk macerate is what I've shown before, what I've shown here. So our bulk macerate is basically what's left over from the macerated residue, which is going to be a mix of microfossils, clumps of kerogen, bits of microbial mats, and just organic fluff. And so the way that I measured the bulk macerate was by just pipetting a drop of all of that goo. And so that's our bulk macerate. It's a mix of all of that, and taking several measurements for each sample and then just averaging what that is. But usually they're quite similar, these values, because they're already averaged once you pipetted those things out. And I'm not sure, it could be that maybe some part of that is lost in maceration, perhaps some kind of fine size fraction. It's been very difficult. So for this, we're using, to minimize contamination, we're not using plastic or nylon filters, we're using metal filters. And filtering through 10 micron meshes has been a nightmare. But I don't think that there's too much of a difference there. So yeah, it could be that some fraction is being lost in the maceration, but I'm not sure what that is at this point.
Okay, we have another comment and then another question. Gregory Italic again says, following on Alex's point, I published evidence of palaeoecology in the Chuar Group last year, so introduction of terrestrial material is a definite possibility. And then we have one from, I'm so sorry if I get your name super wrong, Malgorzata.
Yes.
You're
All
actually
right.
unmuted. Would you like to just ask a question directly?
Yes. So thinking about the taxonomia, if it is a phycoma of prasinophyte, then it is a resting or reproductive stage. In that case, it would not spend much time in the near surface zone. It does not fit very well with your inferences based on carbon delta 13 values.
So some with some prasinophytes, they're also in the plankton, and they can reproduce in the plankton as well. So they're not necessarily just buried in the sediment during reproduction.
I thought that vegetative stages are free-floating and in the water column, but phycoma, which is amorphotite, would be settled. So I'm just asking you, what do you think about this?
Yeah, I think that maybe that's not the case. The case for Simia, because a lot of the prasinophytes kind of dwell in the modern at the surface, and unlike some there are these resting cysts or reproductive cysts of different algae that do sink to the bottom, and they do spend some time in the sediment, sometimes up to like 100 years. But those are most usually spiny forms. But prasinophytes tend to, from modern literature, they do tend to mostly reproduce in the near the surface.
Okay. Thank you.
All right, we have another one from Don Sumner. Thanks for a great talk, Heda. I find the heterogeneity in δ13C really interesting. I found your interpretations consistent with expected variations within a microbial community and the depth variations. There is a lot of local heterogeneity in many, parentheses, all question mark, modern microbial communities. I don't know if that was a question directly, but more of a comment, or you can.
But yeah, like to some extent, we would expect heterogeneity in most modern communities. So I guess why this is fascinating to me and for Precambrian paleontology is that so far most of our information, like paleobiological information, came primarily from morphological inferences about these macrofossils. And so this is kind of our first pass at what are they doing in terms of, where they're taking up their carbon, or what are they doing with their carbon, either metabolically or in terms of their water column habitats. And so I guess this is perhaps kind of our first insight into that heterogeneity and kind of a first step to reconstructing palaeoecology of the early eukaryotes, which is still remains to be explored quite a bit.
So maybe I can add to my question. Would you expect that the same form could show different metabolism depend on redox conditions or other environmental controls?
Yeah, that could definitely happen. I mean, there's so much variation in the modern, where protists are going to be moving diurnally up and down or chasing after some kind of chemokline. And then we also have a lot of kleptoplasty as well, where different protists can just borrow a plastid for a little while, do photosynthesis, and then switch to mixotrophy. And so there is a lot of variation in the modern. So that could also be a contribution to this heterogeneity that we're seeing. And then specifically when it comes to switching metabolisms, it could be that we're seeing also a switch to heterotrophy, but it's not something I would necessarily expect for these microfossils. So for example, there are taxa that are there are taxa that we can we know a little bit more about and are assumed heterotrophs at younger times. For example, like chitinozoans or flask-shaped organisms in the late Ediacaran and so on. We don't see that here, but it is possible that like there are metabolic differences, definitely.
Thank you.
Okay, with that, we're past 9 a.m. And we haven't received any more questions. So that was really great, and we're really appreciating how much this and other talks so far have generated a lot of discussion at the end. So thanks again, Heda, and everybody else. Thank you for showing up and sticking around. Have a good week, and we will see you next week for Dr. Robert Stern.
Thanks.
Oh, come on.